All right. Good morning, everyone. Thank you for coming here. My name is Peter Kwashi. I'm the Deputy Director for Research for this center. And I'd like to start by recognizing a few of our guests, starting with our guest of honor, Professor William Ampofo, the keynote speaker and country director of GIZ, Dr. Dirk Assman. Of course, also our deans, our directors at the university, and country technical lead of the RISA Fund, and representatives from the different pharma and pharma representatives in the room. And then I'll now introduce the chair and our host, the founding director of WACCBIP and the committee, the chair of the NVI Vaccine R&D Committee, Professor Gordon Awandare, to take it from here.
Good morning to you all. Our distinguished guest of honor, Nana Professor William Ampofo. He's a chief who chews guns. So be mindful. Your Excellency, the country director for GIZ, Dr. Dirk. I see that my friend has just arrived. Thank you very much, Farhan. Deans and directors present, deputy directors of WACCBIP, colleagues, good morning to you all. It's a great honor to welcome all of you to WACCBIP this morning and to the University of Ghana, Ghana's premier university, Ghana's hope and glory. I hope you know that this is the #1 university in Ghana. And we set the pace for the others to follow. And in that regard, today we are hosting this very historic forum where we are bringing stakeholders in the vaccine development space in the country to share ideas and discuss where we are as a country in terms of our capacity to make vaccines and where we need to go. Today here we have the entire pipeline from the basic scientist, which is represented by WACCBIP, where you sit. Noguchi people are here, Professor Makusi and others are here. I don't know if KCCR is here. And then all the way to the manufacturing, Farhan is here. And then our development partners, of course, GIZ and others. Our chief himself is here, the National Vaccine Institute, who has been so, strong in pushing for some of the changes that we've been asking for many, many years. Indeed, the establishment of the National Vaccine Institute is a significant step taken by the president of this country to demonstrate the commitment of this country to, make a significant change in our national capacity to make our own vaccines and to be self-reliant. We all remember when COVID struck, the anguish of waiting for crumbs of vaccines from other countries. They would share vaccines with us only after they have taken care of their people, which is logical, right? And they were able to make vaccines in record time because they had existing infrastructure, they had existing facilities, existing technology, and most importantly, existing human resource with the capability to take a new pathogen and turn it around and use their existing structures to make a vaccine within a short period of time. And this is what we have to aspire to be able to do. So we are setting up structures to be able to make vaccines that we routinely use, but also make us stand in readiness to be able to adapt and respond to any emerging pathogens that we may encounter. So ladies and gentlemen, it promises to be an interesting morning. We look forward to the discussions and the engagement, and most importantly, what happens going forward. So at this juncture, it's my honor to introduce my big brother, Nana Chief, CEO of National Vaccine Institute, and overall patriot of Ghana, to come and talk to us. Thank you very much.
Yes, sir.
Yes, sir.
Good morning. It's okay. Good morning, everybody. All protocols observed. As I was coming, I was listening to the radio, and today is Green Ghana Day. So His Excellency the President is being hosted by all the military chiefs, the defense minister, all of them. They are at Bema Camp in the Nicholson Stadium, and they are discussing the planting of trees to combat climate change. So it struck me that one of the places that trees will be planted today is the University of Ghana campus. And I feel very, how do I say? I don't want to use the word welcome, but I feel at home whenever I drive through the campus, because I was in the zoology department. Okay, I did some biochemistry here. And then I lived on campus, and I worked at Noguchi. I still work at Noguchi. I'm still a University of Ghana person. Okay, I'm on some kind of leave. But I was not surprised to hear that the University of Ghana is including a tree planting program. And I think it relates significantly with what we are discussing today. This forum on vaccine research and development, because it is the significant contribution of Noguchi, WACCBIP, the University of Ghana, and other institutions to the COVID-19 effort that gave us the platform to convince the president that we needed to have a special program on vaccine research, development, and manufacturing in Ghana. So again, when he was starting his address, he ended with fellow Ghanaians. And remember, during one of his fellow Ghanaian addresses, he then referred to the need to look at vaccine research and development and manufacturing in Ghana. And he set up a presidential committee, and the presidential committee did an assessment and recommended the establishment of the National Vaccine Institute. So we know the politics in Ghana, So one very significant part of this approach was that the Vaccine Institute and the program for vaccine research, development, and manufacturing should be backed by an act of parliament. So when there's a change in government, the National Vaccine Institute and this particular program is set in stone. It will continue. If somebody wants to change it, they'll have to go to parliament and fight and argue and debate, to change this program. But already the fact that we have gathered here this morning shows the significance of this, important milestone. It was not difficult at all when we were discussing with GIZ colleagues to sponsor such a program this morning here at WACCBIP. This is because research and development is a cornerstone of vaccine manufacturing. During my days working on influenza, I used to be part of a special global advisory committee at the World Health Organization. And one of the things that we discussed was a global plan for the production of influenza vaccines to ensure that should there be a pandemic, there would be enough manufacturing capacity to be able to produce pandemic vaccines from seasonal vaccines. I did remember that at one of the meetings, there was a manufacturer from India who complained that they were making 1 million doses of influenza vaccines and the Indian government was not buying. Fast forward to COVID-19 pandemic, we were crying for vaccines because there were no vaccines initially, to share with us. So clearly, the need for us to develop our own manufacturing capacity, the need for us to be able to do research and development to feed our manufacturing and to be able to make our own vaccines is a major issue. So we will hear later on today the efforts on some of the wonderful work that is going on at Noguchi, at WACCBIP, and also one of the most exciting things are private sector, efforts also in basic sciences that will also enable us to make vaccines. And then also we hear from the regulator. So we have a wonderful program lined up. And I would like to assure you that the National Vaccine Institute will fully support all work that is geared towards the production of candidate vaccines that our private sector can take forward, to actually full-scale and manufacture. When we did the assessment as a presidential committee, we realized that sometimes the impression is given that there is nothing, happening. But actually we realized that there was a lot going on in the research institutions in academia. My own group produces what we call influenza antigens that are circulating and shares with the WHO group that actually influences the decision on the type of influenza vaccine that is made each year. And Ghana was one of the few countries that continued to share influenza antigens during the COVID-19 pandemic, 2019, 2020. So some of the really basic work that we do, in our different programs, does make a significant global effort. You will hear about the malaria antigen discovery and other stuff later on. So we will continue to push government to commit resources. Already some resources have been made available to support research and development. And I think at another forum we'll discuss how that will support, some of you. Those of you looking for jobs, you should be looking out. We'll be advertising soon to pick, those who are suitable to work at the Vaccine Institute. Don't call my phone, just respond to the advert. And if you're suitable, we'll do the assessment and then hopefully we'll make use of your skills. I think the final thing I'd like to mention is the wonderful support from the GIZ and from the European Union. Right from the beginning, they supported the Presidential Committee, and we work very closely with them at the National Vaccine Institute. So as I mentioned, it is very gratifying that we are having this forum with support from the EU through the GIZ. And incidentally, we will be having various programs with the EU and the GIZ going forward. If you've been watching the news, you'd have heard about a big program to support pharmaceutical production and vaccines in Ghana over the next 4 years that the EU is supporting through and also with support from the German government. So much as we will grow our trees, I see you as very important, my skills in handling guns and blank bullets. So I thought he was acting as the MC, but he's asking me to introduce our keynote speaker, Dr. Dirk Abman from the GIZ. He's the new country director, and I've been interacting with him by email and WhatsApp and electronic messages. So today is the first time for us to meet in person, and I'm very happy that he's here with us. I was congratulating him on his wonderful political suit. He's already, learned the customs in Ghana. And so, Dirk, you're welcome to give us the keynote address. Thank you so much.
Well, thanks a lot for the very warm welcome. I liked you started in a very informal way with all protocol observed. I'd like to continue in this way because we are here amongst friends, cooperation partners, I feel at home as well. Well, I didn't study here at the university, but I always at universities I feel at home because before joining GIZ, I worked in the academia for almost a decade. So I'm always very glad to not only to visit universities and research institutions, but also to cooperate with them, because in my opinion as well, research and development cooperation needs to be close together. And of course it's a great honor for me to be here, and I'm pleased to be able to speak to you today. When you believe in scientific evidence, and I guess we all here in the room, we believe in it, it's obvious the topic of vaccine research and vaccine manufacturing that we are discussing today are extremely important. But I'm also pleased to be here because just over a fortnight ago, I also wanted to say a few words during a vehicle handover to the National Vaccine Institute, but flooded roads put a spanner in the works for me and my colleague. So it's all the better that it worked out today. And thanks a lot to WACCBIP and NVI that I can take the opportunity to talk about these important topics, challenges and successes in this area, which by the way, are only possible when partners have a shared vision, face challenges together, and have common goals in mind. If you're from Ghana, from Europe, or from wherever. According to the World Health Organization, over 154 million lives have been saved through global immunization efforts in the last 50 years. 150 million lives. I guess it's very impressive. This translates to saving the lives of 6 people every minute of every year over the last half decade. Significantly, 146 million of these lives saved were infants. Well, these are really huge numbers, but human beings tend to underestimate huge numbers. But it's more than a number. It means that vaccines saved the life of people we love. Your brother, your daughter, your nephew, your wife, or close friends. So without vaccines, also this room would be less occupied today. Look around, some of your neighbors wouldn't be sitting here. The development of vaccines against 14 major diseases like diphtheria, hepatitis, polio, and among others, has also helped to reduce child mortality by 40% worldwide and by more than 50% in Africa. And of course, in addition, pandemics such as the recent COVID pandemic have been kept under control through vaccination. The production of vaccines is therefore of central importance for global health security. But it also offers immense opportunities to economic growth, which we are at GIZ from the German Development Corporation see as an integral part of our work. Vaccine manufacturing creates new jobs. You already heard, so just apply at the NVA. Creates new jobs in the entire value chain from research to manufacturing and generates revenue and foreign exchange that can boost the Ghanaian economy. Countries like India, China, South Korea, Netherlands, Denmark, and others, Germany, are good examples of countries where revenues from the vaccine or pharmaceutical industry have contributed significantly to economic growth. For example, in Germany, the contribution of the pharma industry to the GDP, to the gross domestic product, is about 30 billion US dollar, which is about 40% of the total GDP in Ghana. So there's a huge economic potential. The Ghanaian government has also recognized this and wants to turn Ghana into the vaccine manufacturing hub in the sub-region. But how can vaccine production be advanced in the country? Well, one key area is, of course, research and development. They are at the heart of vaccine production. However, for research institutes such as the WACCBIP to work successfully, considerable investment in laboratory equipment, and personnel, and know-how is required. Therefore, German Development Corporation and the European Union are working with the National Vaccine Institute and research institutes such as the WACCBIP, as well as with the private sector. We hope to focus on in the following areas in the future, in the next year, on connecting research and industry, which is very important as well, and very often quite challenging. Strengthen local producers of pharmaceutical products and qualifying staff in all areas. And the connecting and industry not only nationally but internationally as well. Well, I would like to thank all partners once again for their cooperation. Only together we can succeed in strengthening the Ghanaian vaccine and pharmaceutical industry and truly make the country a global player in this sector. And finally, I'd like to put on the table a question. You mentioned that this university is number one in the country. Last week I visited the KNUST. They mentioned the same. So this will be interesting for me to hear the arguments, but I think Ghana can be proud of having at least these two, and there are more than this, but at least these two excellent universities. Thank you very much.
All right, thank you very much to our distinguished guest for those very lively comments. So we are going to take a short break to have some photographs taken, and then our distinguished country director would have to go to work, so he will leave us whilst we continue the program. So we'll take a picture of the VIPs here first, and then we'll take a group picture with everybody outside. So the rest of you can go to the front of the building. We'll join you there for a group picture shortly. Thank you.
All right. I think that's the good thing of when you don't have political speakers. You can move fast. There's no long speeches. So we are running a bit ahead of schedule, but I think we'll still take advantage of that and continue the schedule. So this is a R&D forum. So we are going to start the academic research output portion of it, which now we are about 35 minutes ahead of time. So we thank our keynotes, our chair and guest of honor for buying us that time. And hopefully the academics are not going to steal too much of that time. Without much ado, I'll call the first speaker, Dr. Yawa Niwe, who will come and present malaria antigen discovery at WACCBIP. That's work that we've been doing over the last 10 or so years.
Okay, good morning. Yeah, so I'll be presenting on the efforts at WACCBIP towards vaccine discovery or vaccine target discovery. And most of the work that we do in the malaria lab focus not only on falciparum but in the spectrum of Plasmodium. So if you hear me present it in the Plasmodium, I mean Falciparum ovale malariae. Okay. So if you're to be talking about malaria vaccine, then we have to look at it in the context of the global vaccine landscape. So if we to look at over the history of human, have we ever been successful in designing vaccine against a eukaryotic pathogen? That is the big question. And have we? No. Over the years, what has been the tools used in eukaryotic pathogens are other biologics that have been employed, but not necessarily vaccine. So that is one of the biggest conundrums that malaria poses. Since it's a eukaryotic pathogen, it is difficult to really be able to design and have very good targets that will be highly immunogenic enough. So looking at the vaccine landscape, it is very evident that anytime there is vaccine, we are able to end disease. So if you look at polio, you look at yellow fever, many of us here have been yellow fever vaccinated, and once you are born, it is given to you. So once the tool is there, it is easy to now use it to curb the onslaughts of the disease. So one particular thing that we do, and what we've been asking basically is, so if you look at malaria as a disease, there are two things. The disease, which is as a result of the pathogen in you, so the reaction that your body poses to it is what we are talking as the disease. But then the pathogen also living in you is another thing altogether. So we have normally what we say anti-infection or it is anti-disease. So these are the angles that we look at it from. And one thing is that to be able to have a good vaccine, it should be stable and it should be effective, and also the half-life, how long it will be able to protect an individual against the pathogen should also be something that is. And in the case of malaria, we know that if we sample us here and we find people with the parasites in them. One thing that is very common is that the parasite from person A might be different from person B. And that is the wisdom of the parasite. We know it as a small organism, but it does not have a brain, but it's very smart. And people believe that it has co-evolved with man for a long time, and therefore it has that memory. So you need something that is strain transcendent. So if we give to person A, it should protect. If we give to person B, it should also protect. So, like we said, it is easy to define, but difficult to really be able to achieve. So that is the initial mindset we need to have. There's no need to deceive yourself that you just get a magic bullet and go. So looking at our parasite life cycle is, first of all, complex. And the other thing that makes it very difficult is that if you look at even the parasite now entering your red blood cell to cause the disease, the number of antigens it uses are a lot. So the simple question is that even if we say that the possible vaccine candidate should be here, which one will you prioritize over the other? And that is one of the biggest problems the field has faced. So if you come to WACCBIP, the approaches we use in vaccine research, we have groups that are looking at the target discovery and also looking at the possibilities of reverse vaccinology. Others are looking at the mechanism by which the possible vaccine will be able to elicit the protection that we want. Others are also looking at now what we call designer vaccines, where you look at possible areas of the antigens that you will be using, and you don't want to use the whole thing. You want to look at specific regions and put them together into a form that will be able to protect. And also others are using in silico methods to identify some of these candidates. One of the key proteins is thought to play a role in our visioning as well. So these are some of the things. So one thing people have thought is that if you look at this antigen, it is very small, so it is easy to produce. So here, what we demonstrate is that he's able to produce the antigen very well. Yeah. The problem with we designing vaccine is not sometimes also about you producing the antigen, but in what form it should be. In the normal system, if we do 1:1, we give you the antigen like single one. One thing is that the immune cells will see it, but will it be able to give the enough focusing so that your immune cells will say that, okay, this is something I must invest in and produce enough antibodies or protective mechanism to. So one thing we've realized from the experiments over the years is that if you immunize with just the full-length RH5, you don't have good amount of protection. So the other things we have always been looking at is that, can we then learn from what viruses use? We know the viruses are particles, but then they have proteins on their surface. So if it's a small particle, you can have as many proteins on the surface. So we have this platform we call viral-like particle. So that is what we designed for this. So what we did is that in this case, we use the hepatitis B surface antigen, but then to be able to get the antigen, which is RH5, to the surface, we have to put a tag we call spike tag. This spike tag will allow it to sit on the viral particle surface so that it's easy to study it. So in essence, if you look at the RH5, this is what it is. If you now attach a spike tag, this is the spike tag, this is we have the capture and then the hepatitis B surface antigen. in a viral construct like this. So if you put this together, you will now get this. So indirectly, we are mimicking the way viruses are able to expose their antigens, which our immune cells see easily. So this is what we did for RH5. And clearly, one thing is that we could purify our viral-like particle here, but then in constituting it, you will have also your protein in units. One thing we could do is that in immunizing mice, we could show clearly that throughout, if you have, this is your hepatitis B surface antigen here, when you immunize, the focus is not on the hepatitis B. We want it to be on the RH5. So after the third dose, you really get comparable amount of antibody that is produced to the antigen. The other thing is that we could show functionality, that these antibodies are not just produced because anything the immune cell sees, it reacts to. But here, the reaction is very specific and very protective. So if you grow the parasite and you now use these antibodies, you see that there is a dose-dependent ability of the antibodies to block the parasite from entering. And this is very, very interesting and very commendable, because hardly do we achieve this level of activity normally in the lab. So this gave us the tool, and we believe that with this tool, we can now go forward and see how. But one particular thing that has been our individual motivation in RH5 is that regarding you and I, if we sample all of us and we ask the simple question, how many of us have antibodies that sees this RH5? Very, very few of us. But then, if there is any one of us here who sees this protein more, you'll be more protected. So the simple question is that if we look at the adults, it's easy. But then when you go to children, the newborn, what will it be? What we have been able to show is that in newborns of parents or mothers who already have malaria, they acquire some level of antibodies to RH5. So the first 6 months level of protection that we always describe, there is a taste of RH5 in the cocktail of antibodies that infant inherits from the mother. So this is one thing that we are now driving down to see how we will be able to design the RH5 vaccine in such a way that it will help not just the normally we look at children under 5, but now also adding the neonates in addition to be able to have very good amount of antibodies in terms of their mothers producing it and then the children inheriting. The other thing we have been doing in addition to the PF-RH5 story is we took advantage of Ghana. There is a unique pattern of malaria. As you move from Accra towards the north, you will realize that for a child in Accra to get malaria, they just need some small amount of parasite, like say 3,000, let's assume. But if you go towards Kintampo, a child will be on the football field playing, but then they have 1 million parasites in them, no sick. Then if you go to the north, which the malaria season is just one, there also you go down a bit. So then looking at these individuals, the simple question is that what allows the child in Kintampo to be able to carry parasite and not fall sick? So we collected samples from Kintampo, collected samples from Accra, and collected samples from Navrongo, and asked the simple question, what is the repertoire of antigens they see? So we got a number of antigens, then we use the criteria of their ability to be used as vaccine candidates. And that allows us to narrow down some of the candidates that I will now be presenting. So here, I'll be presenting one of the candidates we call PF-MAP. What is unique about this PF-MAP is that it is only found in Plasmodium falciparum and Gaboni. So the other parasites, you won't find it inside. But then one thing is that it is also very, very conserved, like the PF-RH5. So what we were able to do is that we could produce the proteins. If you look at the protein structure, it looks very nice. But then the simple question is that, is it stable? That is one of the questions that we've been trying to deal with. The protein has what the central repeat, we call it the armadillo repeats. And one thing with armadillo repeats is that it is a property with proteins that are able to not only be at one location, they are able to shuffle to a number of other locations. So indirectly, it means assuming you are looking at the blast stage parasite, then the forms that the mosquito picks, which we call gametocytes, and then you look at the parasite also in the mosquito. Do we get this protein expressed in that? So what we were able to do is that we could produce monoclonal antibodies here, and we showed that the monoclonal antibodies recognize the antigen nicely. And the other thing is that with the antigen, we could also show here that the antigen is well recognized by the antibodies that you and I acquire just because we live in malaria endemic area. So this is what we succeeded in doing. What we could show is that the PF-MAP antibody recognizes the parasite nicely. And like I mentioned earlier, one property of the armadillo repeat is that it shows different patterns, and that shows that it shuffles from different location to the other. If you look here, you will see something round, round, round. Then when you come here, it's more like dots. It tells you that it moves from being round in nature to being localized to a specific region of the parasite. That is basically what this tells us. The other thing is that if you look at the form of the parasite that is taken by mosquitoes, this antigen is in there. And so far, none of the candidates that we have been using for malaria vaccine exploration have we seen this property. So we think this antigen is unique in its own way, and therefore we will be able to push to see how it will fare in a typical vaccine scenario. We could compare this antigen PF-MAP to already the PF-RH5 that I had mentioned. We see that if you look at, say, among us, many of us will produce antibodies to PF-MAP. So the other thing is the amount of antibodies we produce is more better than RH5. The main thing here is that we could compare the monoclonal antibodies that we produced for PF-MAP, compare it with the monoclonal antibodies available for PF-RH5. And one thing that we can see here is that the green is PF-MAP, and I mean, is it violet or purple? Yes, one of them. Yeah, that is PF-RH5. You will see that the responses are very similar. So with this, what we can confirm from here is that the antibodies are functional. And if you look at, say, the IC50, you will see PF-MAP very, very close to RH5. Therefore, we see it as a promising candidate going forward to pursue. The other thing, just to note, there are 3 parasites that cause malaria dominantly in Ghana. The first, who is the senior prefect, is the falciparum. So anytime you have malaria, the first thing you would think, oh, it should likely be falciparum. You don't know it as falciparum. You say, I have malaria. But likely the biggest suspect is always falciparum. But on the other hand, there are other ones we call Plasmodium malariae and ovale. One thing that is unique about these other species is that this guy, ovale, it is thought that even when you've taken drugs, once they are in your liver, they can decide that, as for me, I'm not going anywhere. I'll just release some few, and the rest will stay. After you've taken the drug and the drug power has waned, then we will come out and see what is happening out there. The other thing with Plasmodium malariae is that it can cause chronic infections, and it has been shown that it is able to drive nephrotic syndrome, and it is able to also cause a number of kidney-related impairment, this chronic infection of P. malariae. But one particular thing we know is that whenever falciparum is there, these guys are quiet. The moment falciparum goes, there is this hypothesis that these guys now begin to come up. So the simple question we're asking here is that with all the interventions, the drugs, everything, focus on falciparum. When we clear falciparum, what will become of these guys? Are we paving way for them to also now assume their senior professorship? That is the question. So here, what we're doing is that with the currently approved two vaccines, the RTSS and then the R21, they target this protein called the CSP. And we know now from the data available that these are able to elicit some level of protection. So, assuming now, if we are to now design vaccine that will target all these three, how will it be like? So that indirectly you are not just selectively eliminating one, but then you will be driving the whole thing, malaria, not malaria from falciparum down. So here we use the PFCSP. We also express PMCSP, POCSP. And clearly, if you use any one of us serum, it recognizes them nicely. So we are thinking that if we could design something for PF, we could also do same for PM and PO and see whether synergistically, if we put them together, we will be able to drive the malaria that we want go down to the barest minimum as possible. So clearly here, what we show is that if you look at different people, there are different levels of reactivity that we see to these antigens. And clearly, this is one thing. Franklin, who is a PhD student in the group, is doing. So if you look at it, if you take people from just community, you see some level of antibodies to all these antigens, clearly. Then if you go to the hospital and find people who are sick, you could see that there are different varying levels of responses to this. So clearly what this tells us is that whether we like it or not, we see the falciparum bit. Now also we have shown that we see the malariae and ovale bit. Therefore, what this now gives us is that it gives us the ability to now go and look at it in this world. Copy is not theft, isn't it? Sometimes it can be, but in this field of science, it is just good to now use the reasoning to now design similar vaccine construct. The other thing is also we could use the VLP system again and see if we could have a single viralized particle that will have PF, PM, and PO on the surface and be able to see the level of protection this will be able to offer us. So in order not to bore you that much, this works that I've presented has been a long-standing work that has been happening in the malaria lab. So the initial work was started with Dr. Emmanuel Amlabu and myself when we arrived in the country as postdocs at the Awandare lab. Dr. Lucas Amingaitogo is more into the genomics because you need to be able to see the genetic diversities of the candidates that you're working with to be able to see that whether this is more conserved, this is more polymorphic, and therefore which ones you'll be able to prioritize. Abdul is joining the group to look at, when we say vaccine, every one of us have vaccine. We've been told that some have 18 years protection, some have 10 years protection. Are they really protecting us that amount of years? And what shows that that is actually happening in us? So that is his work. Hetty is also looking at invasion-related genes. Yao is the immunologist in the group, and when you talk vaccine, you talk immunology. Joe was a postdoc who joined the group, and he also helped in establishing and leading the protein production platform. Professor Samuel Kusi has always been a long-standing collaborator. We've been working, especially on the epitope discovery. He has been leading this for years, and therefore, we've been really enjoying his wisdom. So you will see that we have, this is just a snapshot of the few that we think, but the samples that we receive from the different hospitals, the different patients, the clinicians, everyone is duly acknowledged, and we thank you.
So I think we're still, as an academic, you went over his time slightly, but I think we are still within time. So we can have one or two questions before we move to the next talk.
Second, just a follow-up question. With the IgG levels that you show in the 3 cohorts, how sure are you that they are sort of like, as I said, again, single infection, mixed infections, and the CSP sort of octology, is there sort of like a cross-reactivity that you might be seeing some signals in the 3 cohorts?
Yeah. So malaria, ovale. So one thing that is very clear, and I don't know, but it seems now we are picking a lot of mono infections. The simple question we've been asking is it that we were not looking for them because it is tedious to always look for them, or now we have tools that makes we being able to find them a little easier? So if you go to a typical hospital, now we can see that about 15% of people attending hospital, mostly, will go with co-infection with malaria. Then if you look at mono-infection of malaria, it's around 3 to 5%. So that is what we see. Ovale is around 3 to 5% in there. And we ever had a case of a person who had all the 3 in him. And it was a child. So we are seeing them more now. We don't know whether they are coming up or now the tools that we have is allowing us to pick them up easily. In terms of the antibody, one thing is that the PF-CSP has the NAND repeats. One thing is that if you look at the repeat regions of PM-PO, the signatures are different. So if you do a structural modeling, and you want to see the level of alignment between these 3, it is very weak. And also, even if you look at amino acid alignment, it's around 30 to 40%. So one thing we have been doing is now looking at cross-reactivity. So one thing we believe is that there is not so much of cross-reactivity, but then the other thing is the level of cross-reactivity based on amino acid or it's conformational epitope based. That is the other thing Franklin is now driving home on. And we believe by the end of the year, maybe he should have a few papers out on this.
Okay, I think Nana Prof has a question, and then after that, Farhan.
So when is your candidate going to get clinical trials?
No, so one thing is we now have the construct, the VLP constructs. So we are first of all, you have to look at the construct that you have. No, the construct, how much antibodies it will elicit. So that is the first thing that we want to do. Okay, so for PFMAP and PFRH5, if we have the money, yes, if we have the money, in 3 years, we could begin to test it. Yeah, oh, because as a stand, I don't have the money.
Okay. And I think Fahan had a question also.
Okay.
Okay, the question is like, when we talk about malaria, and we are talking about a lot of discussion is happening in malaria, the problem was like tissue-sensitive immunity was always a problem, because generalized immunity is easy to achieve, and tissue-sensitive immunity is always difficult to achieve. Yeah. Now you have achieved it. And another problem is like, I will echo Professor Ampofo, so that first of all is like lab, when you are developing a titer in lab and shaking flask, it is very easy and it goes very cool. But when it goes to the scale up, yeah, the problem which comes in it, that how you control the contamination and how you pure that characterization of that product is very well done unless it reaches to the clinical trial level. So no doubt you do not have resources right now. So have you tried on a liter basis that, okay, the scale up has gone perfect, and you are very confident that when you have the funds available that you can go for the clinical trial?
For the VLP constitution as a vehicle, yes, that is very, so I mean, just to piggy on what you mentioned, I think that is why we are solidly happy for the initiative of the National Vaccine Institute to have a pilot production lab. So like this, we can easily go, scale it up, say to, now we work within 10 liter range. If we have, say, 50 liters, how much will we be able to get? And that will be a very good thing to do. So we're still open. And like you will see in my acknowledgement, we're still working with Oxford, the Jenner Institute, a group there which is helping us to see how much we will be able to scale up.
Then also, sorry, Doctor, there's another a discussion which is going on right now is antibody vaccine conjugates, because vaccine is available, R21, yeah, it's showing good results. Then antibody you are developing. So are you also working on the antibody vaccine conjugate, because that is the future which, yeah, is basically right now, yeah, predicted in the next 10 years.
Yeah, yeah. I mean,
so have you started work, and what is your work right now there?
So you will see the approach we use to many of the antigens that we're looking at is we don't just look at the antigen, we try to also look at producing monoclonal antibodies to it. The specificity of the monoclonal antibodies is something, and the functionality, that is where we're looking at. So once we are able to have a very, very functional monoclonal antibodies, then you can now look at the antigen-antibody conjugates and then be able, and it is actually, there are new technologies that are also now evolving on how to really have this constituted and being stable also.
So that's what I was kind of alluding to. You guys do a lot of basic science, yeah, stuff. You showed us the epitopes, you showed us the immunodominant regions, etc. We want to push you now to have a targeted, kind of pipeline, yeah, whether it's the antigens that could be candidate, yeah, vaccines, or whether it could be monoclonals that could also be used for therapy. So we want to move away from the traditional, yeah, basic science, or you can continue the traditional basic science, but we want it to be focused, yeah, so that then we can then put in resources that will enable us to say that, okay, after 2 years of supporting basic science research, we now have a pipeline of particular products which we can move forward, from discovery actually now to commercial, feasibility or viability. That would be very important to have the resources allocated to support you. So we need you to develop those plans in addition to looking at exciting or innovative basic science that, answers a lot of the questions, but also looks forward to the end product. That is very critical, for us to go forward. Thank you.
Yeah, thank you.
All right. Thank you, Dr. Aniwe. So in designing the schedule and the speakers, we also wanted to talk about one of the reasons why we are here today, as our guest of honor intimated before, is that of the COVID-19 pandemic, which brought a whole host of issues regarding vaccination, and also coincided with a lot of mis and disinformation. So one thing that we are also doing along the lines here is a project that ultimately leads to the production of monoclonal antibodies from patients. So Dr. Irene Owusu is going to start us off from the beginning and head towards there. So I'd like to call her to come and give her presentation.
All right, good morning, everyone. I am Irene Owusu Amwako, and I'm a virologist, but here at WACCBIP I work as a postdoctoral fellow. And today, I'll be sharing with you work that is ongoing on the expression and durability of multivariant SARS-CoV-2 neutralizing antibodies after people have been exposed to the vaccine, and then we'll have some cases as well. This doesn't work. So last year, sometime in March, WHO declared the end of the COVID-19 pandemic. And, excuse me, sorry, this is just not working. So we were excited to hear that the WHO had ended or declared the end of the pandemic. But then it's also important for us to keep in mind that the end of the pandemic does not mean that the virus has been eliminated or eradicated from the system, meaning that as long as there are people who are susceptible to infection and then the virus is still in circulation, the severe effects or the risk of getting COVID-19 and then certain outcomes is still possible. Now, we all agree that during the pandemic, the introduction and administration of the vaccine played an important role in helping to reduce the severity of the infection. But so far, we don't know for how long this protection from the vaccine and even exposure to SARS-CoV-2 would protect people. And so it's important for us to know what the durability and then the breadth of vaccine immune response after exposure to the antigen is. That's why our projects worked on. We try to look at neutralizing antibodies that people who have been exposed to SARS-CoV-2 antigen would get, and for how long the neutralizing antibodies would stay in these people to protect them. This information is always important to also inform vaccine schedules and how much antigen or vaccine doses should be given. So for a vaccination to be successful, we all know that among other things, two main things are what we'll look at. There should be there should be memory cells produced, or the vaccine should be able to induce the recipient to produce memory cells, and also the antibodies produced should be able to neutralize the pathogen. Now, these neutralizing antibodies are able to, okay, thank you. So these neutralizing antibodies are able to reduce or prevent viral infection by various mechanisms. They can either prevent the attachment of the virus to the cells for entry, or once the virus has attached, they're able to prevent entry or penetration of the virus. And in other instances, the neutralizing antibodies interfere with the uncoating of the virus for the replication to occur. Now, in all these things, neutralizing antibodies always recognize the epitopes of glycoproteins or surface proteins of the virus to be able to interact and then inhibit their action. So our main project was to first identify plasma from patients who had been vaccinated or had the infection, and identify certain plasma samples that could be able to neutralize different variants of SARS-CoV-2. And to do this, we used the live virus neutralizing assay. Here at WACCBIP in our lab, we have various systems for testing for neutralizing antibodies. We have the pseudoviral system and the live virus system, but data I'll be showing you today are data from the live virus system. So for the live virus system, once we get samples, we serially dilute the plasma samples, then incubate them with a known amount of virus, incubate for an hour before we put them on ViroCells. We use a ViroCell system, and we do the infection for up to 3 days, and after that we stain and look for where there's virus infection or not. So where there is no virus infection, like the deep blue wells, that is where we know that there are antibodies or neutralizing capacity of these plasma samples that prevent the virus from infecting the cells. And then we quantify and then get the neutralizing titer. So it's a straightforward method. Compared to the pseudoviral system, this live viral system is more cheaper, and it's easier for us to do here in terms of logistics and then costs. So for this part where we wanted to identify plasma samples that have neutralizing antibodies, we collected samples from patients who had COVID-19. And we sampled across the different waves of SARS-CoV-2 from wave 1 through to wave 5. Now, almost 200 samples have been tested so far, but then in our biobank, we have thousands of these plasma samples that we are yet to test. And of these, majority of them were unvaccinated, while a few vaccinated. And that is how I'll be analyzing the data to try and make meaning of. So speaking of the waves, I grouped the samples in waves based on how the times that they were collected, okay? And so for the period here at WACCBIP, we were able to find that during wave 1, that span between January 2020 and November 2020, the dominant variant was B1.1, and then the peak period was in July 2020, and it goes for the others. So you find that wave 2, the dominant variant was Alpha, and then wave 3 was Delta, then 4 and 5 were Omicron, but different lineages of Omicron. So this is how I grouped my samples according to the times that they were collected. And now for this project, I've tested so far just 4 variants. So the wild type that has the D614G mutation, and then we have Beta, Delta, and Omicron. Now you realize I put Beta in color because we did not record Beta in Ghana, but then I'm testing for Beta or neutralizing antibodies against Beta anyway. And looking at certain spike amino acid substitutions, as I mentioned, the neutralizing antibodies would normally interact with surface proteins. And in the case of SARS-CoV-2, it's the spike protein that is one of the important proteins for immunogenicity. And so looking at the spike protein of these 3 variants that I use, how they differ from the wild type, you find that the beta has just 15 substitutions, while the Omicron has 58. So we see, which is more logical, that as the new mutants or new variants come up, they have more substitutions or more mutations from the original. And so in this case, beta will be more closely, will look more like the wild type. It's closely to the wild type than Omicron that came later. And so this is something we'll also have to keep in mind as I show the data. And so when we run the live virus neutralizing assays for the different samples against the different SARS-CoV-2 variants, I found that most of the donors that we had were able to produce neutralizing antibodies against these 4 variants. So we were able to identify plasma samples that had cross-variant neutralization. And across also the different waves. So you see that as the waves increase, there will be an increase in level of neutralizing titers, which is, it makes sense because as the waves increase, people are more exposed to more of the variants, and so there's continuous antigen stimulation in logic. But then one thing I realized that struck me and then that we want to look into is for wave 2, we find that, so logically we would expect that from wave 1 to wave 2 there will be an increase. Then from wave 2 to wave 3 there will be an increase. But it doesn't happen like that. There is a dip in wave 2 and before the increase starts again. So you realize that people who were exposed to the variants in wave 2 might not have better neutralizing capacity as people who are exposed to the other waves, or to the variants circulating in the other waves, okay? And so the question is that, is it true that variants in wave 2 are likely to elicit poor neutralizing antibody capacity? Now, to do this, we would want to even look into testing neutralizing antibody levels against the variants that were circulating during the wave 2 pandemic wave. And these variants were alpha, eta, and then B1.318. These were the most dominant variants circulating in there. So my question, and for my next step, is to look for whether these other antigens or even these virus variants are able to elicit enough neutralizing antibody capacity. Okay. So knowing that we have patients who are able to produce neutralizing antibodies against different variants of SARS-CoV-2, the question was whether vaccination will play a role. So now, before I showed you samples from all the waves put together, there were a mixture of people who were vaccinated and unvaccinated. So I decided to separate that and then see, it would be a good question when we want to formulate new vaccines against SARS-CoV-2 and then want to decide on what variants to put in there if there is a need. I would think that beta, if tested under other conditions, would be a good candidate, since even for all the waves and then against the different people, there was high levels of neutralizing titer against beta. And so now we know that vaccination is important for neutralizing antibody production. Now, will vaccination also be important for the wide range of cross-neutralization that we see? Because what I've shown with the chi-square analysis is that there's just an association. But then for the different variants, would vaccination help? Now, this is just preliminary data. In the cost to produce neutralizing antibodies in the lab, so monoclonal antibodies, we selected some of the samples. So some of the plasma that express high levels of neutralizing antibodies, we selected their PBMCs for single-cell sequencing. So this is preliminary data done by Kessie, one of Dr. Kwashi's PhD students. So we wanted, after analyzing the B-cell receptors, or after sequencing, we realized that between vaccinated and unvaccinated persons, there was a high B-cell clone, unique B-cell clone in the vaccinated persons than unvaccinated, meaning that the B cells that are produced in the vaccinated persons have wider ranges that are likely to produce antibodies that can neutralize or work against different variants if possible. So this is just preliminary data, but then it's pointing to the fact that vaccination may be a factor towards producing cross-neutralizing antibodies. We tried to test this between symptomatic and asymptomatics, which was the focus of Kess's work, but then there was no difference. And then for the different waves, there's a slightly increasing level of B cell clones as the waves go. But then you realize that as the waves increase, more people were vaccinated. So it still points towards vaccination and antibody clone expansion. B cell clone expansion. Now that we know that we have cross-neutralizing, or patients produce cross-neutralizing antibodies against different variants, we want to know what the durability is. So we're able to produce these neutralizing antibodies that neutralize different variants. And for how long can the body hold these neutralizing antibodies for them to be potent? That is what durability is. So we have a project that is ongoing. So far we've collected samples from 21 donors. So this is a longitudinal study, unlike the previous one that was just a cross-sectional sample collection. So 18 of the participants were from COVID-19 patients, and then 22 were from vaccinees. And so for the vaccinees, we followed them through for 8 series of samples, while for the COVID-19 patients, we took just 6 series, up to 44 weeks. And all these samples were collected just recently during the Omicron stage, or the Omicron wave, so we don't have waves for earlier people. So what happened? So the preliminary data that we've been able to analyze shows that neutralizing antibodies can persist for up to 20 weeks in people who are vaccinated and infected. Now you find that the people in the cases in the red boxes are those that have not been infected, and so you'd see less neutralizing antibody across the series. Where the crosses are where we weren't able to get samples within the series for a particular patient. But you find that generally there is an increasing level up to week 20. That's when most of the times the neutralizing antibody levels will wane down. This could mean a lot of things, but then we can only say more after we've analyzed most of the data, because in the course of these series 2, people can be re-infected or exposed to the virus that can also affect or boost the levels of neutralizing antibodies. But generally, after sufficient exposure to the antigen, it takes about at least 20 weeks for persons to hold the neutralizing antibodies before they start waning off. Now, for people who are not infected or did not show symptoms, but then came in for just vaccination, at day 0 before vaccination, so at this point they didn't have any vaccines taken, you find that it increases with time after they've taken the vaccine. But here we've only been able to analyze up to 8 weeks, and we find that generally people would have persistent neutralizing antibodies against Beta and Omicron especially for up to 8 weeks. Now we are yet to analyze samples for up to 52 weeks and also test more samples and see what the trend truly looks like. But then my colleague after me, Eliza, will talk more about these series of antibodies or patients who have been followed for a longer time and even in a more larger cohort. So in summary, I believe I have I've shown that neutralizing antibody assessment is neutralizing antibody titers is a good assessment for vaccination, because I've shown that vaccinated persons are more likely to express neutralizing antibodies. And so when we want to test the efficacy of a vaccine, it would be nice for us to use neutralizing antibody as one of the measures as well. And when we take a neutralizing antibody against SARS-CoV-2, whatever variant that infected, it's likely that that antibody can, so when we take plasma after a SARS-CoV-2 infection, it's likely that that plasma can neutralize more than one variant of SARS-CoV-2. So we can have broad neutralization. And then the neutralizing antibodies can persist for at least 20 weeks after exposure, and vaccination is able to increase unique B-cell clones, which is likely linked to broad neutralization. And on this note, I'd like to thank my lab, the Kwashi lab, that is led by Peter Kwashi, and then these members in the lab who contributed directly to this work. And our study participants and medical centers, especially the GIZ Medical Center, and then the Greater Accra Regional Hospital, and our partners from Glasgow, who did the single cell sequencing data, for which we had the B-cell clone data from. And of course, these funders, the Wellcome Trust especially, and the Melinda and Bill Gates, and FCDO, that funded sample collection as well as all these ones. So on this note, I'd like to thank you and ask for questions. Thank you.
And we also like to thank Nana Prof. Ampofu, because a lot of the early sample collection particularly was with his guidance.
Hello.
And also Prof. Awandari.
One web. I use like one web.
All right. Any questions?
Hello. Thank you very much for the brilliant presentation. As a public health specialist, I'm always inclined to knowing what actually does this imply for the population. I see that you've chosen to take a longitudinal study to be able to observe if the patients or if the people still have neutralizing antibodies by the 52 weeks. But do you think you can actually use a predictive modeling in order to evaluate if they still need vaccination? And then in addition, I'm seeing that you took actually patients or people who were vaccinated. Was it once, twice? Do these people actually still need boosters? Or are they, or were these people, did these people have boosters? What is the implication of boosters in this population? And I mean, I have a lot of questions, but it's mainly, do we still need to vaccinate people? And do we still need to have much more boosters? And what does it imply for the patient?
Okay.
From your preliminary data.
Yes. So yeah, basically what you want to know is whether people need boosters to be fully protected from SARS-CoV-2 infection or the severe effects of SARS-CoV-2, because we would always be exposed. So from the data so far, if what our preliminary data is showing is true, that after 20 weeks or whatever, after X number of weeks, if we have waned down neutralizing antibodies, and if we are hinging only on neutralizing antibodies, and we find that after X number of weeks it's waned down, and we are starting to find, we should be able to predict, if we have a large sample size, to predict that if after X number of weeks there is a decrease in neutralizing antibodies, then we should be expecting a spike or an increase in cases again, if the neutralizing antibodies will be a good measure for protection. You understand? So we can predict that after December 2024, there should be an increase in SARS-CoV-2 cases again, because at that time, almost everybody who had taken the vaccine would have waned off. So it will be a good predictive module so that we can prepare that if that happens, what can we do? If we boost, what does it happen again? If we give them another boost or introduce them to the antigen or however. So it's a good thing to prepare for the future. If after the end of the time it doesn't happen, we still were prepared. But then it's always a way to think forward and then to predict. Otherwise, there's no way to know what to do. Yes. Have I answered? Yes.
Let me add a little bit. So the other thing is that based on the sample sizes that we have, it's very hard to build a predictive model. But I think it's quite clear that if you are not vaccinated, you would have vaccination would add to the protection that you have from a natural infection. So if you're not vaccinated, definitely being vaccinated will still give you an added level of protection and durability. What is becoming clearer now from the cross-reactivity is that maybe boosting with different variants may not be as important as the frequency of boosting.
So my question has to do with the neutralization data you showed. It was hard to see because you had lots of points smashed down on your axis. So what I was interested in, I don't know if you can pull it up, but are the outliers driving your differences, or do you have, because it looked like you had a group of people who made no neutralization at all. Mm-hmm. So have you looked to see what those people are? Why is it that they were not able to neutralize? Because you seem to have two groups. You have some that are very high neutralizers and then some that don't neutralize at all. And yet you've grouped them together, right? So I wonder if you're actually conflating. Maybe there are a group of people who don't neutralize at all, and the question is why? Were they not exposed? Did they not make a response? Did the vaccine not work in them? Versus the people who neutralize really well. Because right now you have almost like a bimodal population. So I'm just curious if you tease that apart a bit.
So teasing them apart, most of them were unvaccinated people, but then all of them were exposed because they were COVID-19 patients. They tested for COVID and then were part. So the other thing too is that when we ran a total antibody, I should have showed that data. For total antibodies, some of them had total antibody levels, okay? But then when we do the neutralizing antibody titers, they don't have at all. So they were exposed, but then their antibodies that they produced were not neutralizing. And so we may want to look deep into what the factors are. And that is what the single-cell sequencing data also we are trying to look at, because we took some of the patients that did not show neutralization at all to sequence their B-cell receptors to see what it looks like. Yeah,
I think it would be interesting to look at, because, and maybe display it differently, because right now the axis you're using, most of your points are very low to the axis. Mm-hmm. So it would be helpful to see if there is actually more of a spread. Okay. And then if there are a group of people who do not respond at all, who are unable to neutralize, even though they have antibodies, that's actually really interesting, right? It's like you have antibodies, but it's not working. Yeah. And what is it about those people that the antibodies don't work?
Right. All right.
All right, thank you, Irene. So, the other thing is, what we are going to have here is, because we have a company that is near and dear to us at WACCBIP, which is a private company and also doing a very similar study focused on vaccine responses to SARS-CoV-2 vaccination. We wanted to follow on with that, and so there will be some similarities and some differences to what Dr. Alicia from Yamachi Biotech is going to present to us. I'm not going to mention the full name, so I'm sure you'll mention it when you're presenting.
Yes, thank you very much. Yeah, so I'm Elise Marie Kwesi-Malipaard, Dutch by the Canadian. Anyway, so I work at Yamachi as a research scientist, and at Yamachi our goal is to transform lives through innovative and inclusive science and technology. So Yamachi is Africa's first cancer biotech research company, launched 2021, headquartered here in Ghana, but also with labs in Nairobi, Kenya, and an office in Washington, D.C. And we work in the whole of Africa. So we have partnerships with hospitals, research centers in 9 African countries. And so the heritage study took place in the Greater Accra region. We included 301 adults from 4 different locations where they got the vaccine. So at Dabra Polyclinic, Greater Accra Regional Hospital, Kolebu, and University of Ghana Hospital. And so 7 time points. So in total we collected over 15,000 samples. So the samples we collected were mainly oropharyngeal swabs to look for COVID-19 positivity and blood samples to do the full immune investigations. So you can imagine that is a lot of data that you generate with so many time points. Participants were also followed up in between to ask them how they were doing, COVID-19 symptoms. So we have managed all this data in Uvosio. Uvosio is the data management system developed by Yamachi, where we can manage all this data in a structured way, pull it out easily so we can do our analysis. So the demographics of our participants. So we had 301 participants, around 60% of them male, most participants quite young, half of them between 20 and 29 years old. We asked them whether it's COVID-19 symptoms at baseline, and around 25% reported 1 or more COVID-19 symptoms. We asked for comorbidities, autoimmune disease, and as I mentioned, so 45% got AstraZeneca and 55% got the Pfizer vaccine. Yeah, so one of the first things we checked was COVID-19 positivity during the course of the study. So this was done by RT-PCR. And as you can see, at baseline, around 20% of the participants were positive for COVID-19. This 20% was roughly maintained for the first 3 weeks. And after that, we saw a decline in COVID-19 positivity, which I think is a very good thing you want to see when you do a vaccine study. So when we then look at how does our data compare to the situation here in Ghana, so these orange bars is COVID-19 positivity in the heritage study. So, and then in blue is the new cases in Ghana, and in red is the new deaths in Ghana due to COVID-19. And you can see that actually our data quite nicely follows the national trends here in Ghana. Maybe also good to notice this pink line is the people vaccinated, and our study started December 2021 when vaccination in Ghana was still very low. So when we compare our trends with the global trends for COVID-19 cases and deaths, there's not so much correlation, especially not for this last peak. So yeah, so one of the first analysis we did was to see, okay, if you do a proper modeling, do we indeed see that when you get the vaccine, the chances of being positive for COVID-19 reduce? And that is indeed what you can see, that in the first 2 weeks, participants are more likely to be COVID-19 positive, and after that, less likely to have COVID-19. And we also looked at other factors that might be associated with COVID-19 positivity at baseline. And a few of the factors that came out were those who had formal employment with a higher odds of being COVID-19 positive, those with comorbidities, and those with COVID-19 symptoms. I think that one might not be a very surprising finding, but I think it shows that the data is reliable. And we also saw that those with autoimmune disease, here have a reduced likeliness of being COVID-19 positive, and that might feel a bit counterintuitive, but what we think is, and it's also literature supporting that those who know they are vulnerable, they adhere better to all the measures like wearing of nose mask, washing of hands, and that they better protect themselves. So now I would like to take you to the serological analysis. Yeah, so I would like to thank the previous speaker for introducing neutralizing antibody titers, the spike protein, all of that already, because as Dr. Kwashi mentioned, we are doing similar things. So the first thing we did was to look at antibodies against the spike protein and against nucleocapsids. And what you can notice at that baseline, there are already quite high antibody titers for both anti-spike and anti-nucleocapsids. Especially anti-nucleocapsids, it indicates it was high previous exposure, natural exposure to SARS-CoV-2 in this cohort. And keep in mind, this cohort started December 2021, so COVID had already been around for some time. I also want to draw your attention to this group. These are like 45 participants who were anti-nucleocapsid negative at baseline, so they're most likely SARS-CoV-2 naive. So then when we look at our neutralization antibody titers, also using a live virus neutralization assay, and thanks again to the first speaker for explaining that in depth. So what we see here is that basically for all the variants at baseline, the participants already had quite high titers, and they were even further boosted upon vaccination, and then over the 12-month period remained quite stable. And then we wanted to go a bit more into this, see how does natural exposure also affect this. So we divided a population into those who were anti-nucleocapsid negative, so previously not exposed, SARS-CoV-2 naive, and those who were previously exposed. And then we just look at the median antibody titers for the different subsets. So the solid line are those who were SARS-CoV-2 naive, and you can see they start off with much lower titers as those who had previous exposure. And then they see the result of the vaccine, they get boosted, and they maintain their antibody titers over the 12-month period. Those who had already previous exposure, they started, of course, higher, and they also benefit from the vaccine because their titers are boosted even higher, and then there's mild waning for most of them, but still remain often higher than those who were previously naive. Except for one exception, which was Omicron. And here you can see that those that previously exposed, they start high, the titers get boosted, and they remain stable. And for those that were SARS-CoV-2 naive, they get a boost from the vaccine, and then they go up to similar levels as those who already had previous exposure. And so this study was mainly in 2022, starting end of 2021. So Omicron was also the most prevalent strain during the time to study. So what we think that we see here is a very nice example of hybrid immunization, where we have the combination of natural exposure and vaccination, which we know that together gives even a better immune response than any of them separately. So what we also did in the legacy study is our study is designed in a parallel design to the legacy study in the UK, and this enables us to compare both studies and see if there are geographical differences in the vaccine response. It should be good to keep in mind that legacy did start around a year earlier than heritage. So the variants that were going around during the legacy first year were different variants than going around during heritage. And I think one of the first things you will notice is that basically for all variants at all time points, the neutralizing antibody titers in the UK in the legacy study are lower than in the heritage study. And especially for Delta and Omicron at baseline and legacy. So this was January 2021. Those variants are not around yet, and we also see that the legacy participants do not have neutralizing antibodies against them at baseline. So we were then very curious to see how does the legacy data from the UK now compare to our different subsets based on their previous exposure. Yes. So we have again the same graph. So here those heritage previously exposed, solid line heritage previously naive, and then the small dashed line is the legacy data. And we find it quite interesting to see that legacy and those in Ghana that were not previously exposed to SARS-CoV-2, they seem to show, in general, like similar trends. They start off low and then they plateau at a certain level, except again for Omicron BA2, where we see it in legacy. And remember, legacy, this was still in 2021, so Omicron had not much arrived yet. So legacy plateaued, and whereas in our heritage study, we see that both, because natural hybrid immunity, they get higher titers. So I think that, in summary, we can show several things. We see that in the previously exposed, in a population of high previous exposure, giving the vaccine still boosts the immune response. We also see that hybrid immunity, a combination of previous exposure and vaccination, even further boosts the immune response. And we have a study in a parallel design to another study in the UK. So it also enables us to look at, to compare the vaccine response in different geographic locations to see, look for molecular cellular mechanisms underlying differences. So our future work will focus on looking at the cellular immune response to the SARS-CoV-2 vaccine in the legacy studies among the Ghanaian vaccinees and compare those to the UK vaccinees. We want to do ex vivo immunophenotyping, transcriptomic profiling, and also single-cell sequencing to look for specific T-cell and B-cell clones that are relevant. And we also want to go and look at how the host genetic diversity affects innate immune response. Furthermore, we are part of the WWC consortium, which consists of 3 studies. So the heritage study here in Ghana with GIZ and WACCBIP, the legacy study in the UK with the Francis Crick London School of Hygiene and Medicine and University College London Hospital, and the Windfall study with the University of the West Indies, which will allow us to do even more comparisons between different geographic locations. And then I would like to thank, first of all, the participants in the heritage study, everyone at GIZ, WACCBIP, the Francis Crick Institute who have been involved in this work, the Ghana Health Service, and the Bill and Melinda Gates Foundation for sponsoring this study. And I'm happy to take any questions.
Thank you, Alicia. Any questions for Alicia? Charles.
I was just wondering, with the sero response that you saw comparing spike and the nucleocapsid sort of proteins, I just realized that you didn't see much in the nucleocapsid, right? But did you do some sort of like a subgroup analysis in certain age group to see whether you have some responses in certain age groups?
For age groups? No, we have not done that. We have just looked at the total of participants.
It will be interesting to see how the age groups fare in terms of the responses.
We are vaccinating adults, right?
Yeah, they're all adults. Everyone is 18 and above.
Yeah, so you could do maybe the younger age groups, and then maybe middle age, and then those who are a bit older. It's just to see how, because immunity is sort of associated with age. But then also a follow-up question would be, when you follow up the participant, did you also look at other, I think your listing was the end outcome was primary, was it seropositivity or PCR positivity?
The COVID-19 positivity is by PCR.
By PCR, okay.
Oh, I thought you had a question. Okay, so let me. Okay, so let's give another round of applause. So we've managed to catch up, and I think a little bit early on time again. These are the three sort of basic science presentations we wanted to give to show a flavor of what we're doing here in terms of the antigens that we think are a little bit close and have the potential to be developed for the clinic. And then you see that the work presented by Irene and Alicia have sort of overlaps and feed off each other. Irene's is more related to the natural infection and the impact of vaccination. And the outcome of that project would, like I mentioned, would be the monoclonals that would try and test and use as reagents, and also potentially see the effects as reagents or therapeutics, or whether they have functionality in diagnostics. And then the vaccine response study will sort of bridge that and build on that. So we thought it would be good to showcase Yamachi, since we are collaborating on that study as well. What, I'm not sure if he managed to come here, what we wanted to close this out before the general discussion would be to have a presentation by the regulator. But I understand that Dr. Nkansa just arrived in the country last night and has not been able to make it here. So I think we will take our lunch and then come back here for the general discussion, which will involve our pharma partners, DEK, Atlantic Life Sciences, Yamachi, and some of the speakers. So the lunch for the invited guests will be on the first floor of the biochemistry building, and then for everyone else on the second floor. So let's try and be back here by 1 o'clock for the general discussion, and then we close out after that. Unless if there's any questions. So we'll come back here after lunch. At 1 o'clock. And we'll take attendance. Okay. I believe we are mostly back now. We just wait for the high. Yeah. So for this next phase, I'd like to invite representatives from DEK Vaccines, Mr. Farhan, to the podium, along with the representative from Atlantic Life Sciences, and then Yamachi Biotech. So we'll give a few minutes to present your company, and we'll discuss a little bit about the work that we've done here, that we've shown here, and where we think we are, what are the positives, and how is it that we can facilitate more academic industrial partnerships, and where are we going in terms of vaccine R&D in Ghana from some of the ideas that you've seen here. So I'll start from my right, going to the left. And I believe, Fan, you have a video to show us? Okay. So they have the video? And I'd like everyone to fully introduce themselves first before we go on. Switch it on. I think they have to give you power. Can you power the mics? Try again. Okay.
Thank you very much. My name is Farhan Ahmed Khan. I am the project manager for DEK Vaccines Limited. DEK Vaccines Limited, almost all of us know that DEK is the upcoming vaccine manufacturing factory in Accra, Ghana. Before I go ahead, I have a small video of the company, so I want to show so that we have a fair idea of the company, and then we move ahead from there. So am I allowed? So please play the video which I have given you, IT team. So the groundbreaking happened in last year,
located in Accra, Ghana. We are thankful to Almighty that COVID-19 pandemic is over. World has learned many lessons from COVID-19 pandemic.
Vaccine nationalism that was played out by the developed world with the rollout of COVID-19 vaccines meant that we had to take urgent, critical steps towards making sure that never again would we be victims.
It was an eye-opener to African leaders, where His Excellency Nana Akufo-Addo, the President of Ghana, expressed his vision that Africans cannot depend more on imported vaccines. There must be an African solution for an African problem. DEK is rapidly completing the civil construction because of His Excellency vision and initiative to set up Presidential Vaccines Manufacturing Committee, who developed the business plan for vaccines manufacturing in Ghana. Once proven that Ghana is the most appropriate country in West Africa to have vaccines manufacturing factory, VMC invited private sector to join hands to build the mega project to manufacture vaccines in Ghana. DEK Vaccines Limited was officially selected to lead the vaccines manufacturing in Ghana. Kina Pharma Limited, the only shareholder of DEK Vaccines Limited, stepped ahead to make His Excellency the President of Ghana vision into reality. Dr. Kofi Nsiapoku, the chairman of the Kina Group Limited and managing director of DEK, stands strong to have the pride of developing DEK Vaccines Limited to develop the health security in Africa. DEK Vaccines Limited is a $175 million investment to manufacture 600 million doses of various vaccines annually. Government of Ghana had passed the National Vaccines Institute law in the Constitution of Ghana. This has exhibited a lot of confidence in investors and shown the practical approach of leaders to Ghana to save African children from infectious diseases. Government of Ghana fully support DEK Vaccines Limited by issuing an off-take guarantee to procure the vaccines from DEK once DEK start manufacturing vaccines of WHO quality standards. FDA Ghana registered for in Africa. It is also an example of long and productive public support to the private sector. DEK, upon completion, is ready to manufacture malaria vaccine, oral cholera vaccine, hexavalent vaccine, HPV vaccine, typhoid vaccine, pneumococcal vaccine, rotavirus vaccine, hepatitis B vaccine, yellow fever vaccine, BCG vaccine, vaccines on Chadoc's platform, measles rubella vaccine, meningococcal vaccine. Tech transfer of OCV to DEK is aimed at increasing global vaccine production capacity by 90 million vaccine doses of oral cholera vaccine under tech transfer from EU biologics per annum and reducing disparities in vaccine access for African children. DEK tech transfer for oral cholera vaccine and other vaccines will add 600 million doses of vaccines in global vaccine production and supplies. DEK's 600 million doses per annum production capacity is a strong step towards health security of Africans and preparedness for the next pandemic. DEK Vaccines Limited, made in Africa, for Africa.
Thank you very much. Vaccines are basically, vaccines have saved a lot of lives of human beings. Without vaccines, number one, the health security is vulnerable, and this was basically come down to the mind of African leaders, and they realized during COVID that we should have a vaccine manufacturing factory. DK, the building you have seen under construction is almost complete now. The first building is complete, and we are going to produce vaccines in 2026. 2025 we are installing the machines, and 2026 we will be doing the validation. After doing validation, then in 27 we will go hopefully toward the registration with NRA, which is FDA Ghana. So we are on the track, and initially we will be launching maybe like 2 or 3 vaccines. You have seen the list of vaccines that cannot be possible to be launched in 1 year. So this is a vaccine portfolio which maybe you're seeing in next 10 years in DEK once it is started producing. So the objective of today's discussion was like, okay, to basically give you a fair idea where DEK is standing, as well as what is the vision of DEK. Fill and finish. Fill and finish is not a sustainable business model in vaccine manufacturing. You should have your own IP, and you should have the drug substance manufacturing with collaboration with some of the companies. So DEK has worked a lot. There are basically discussions, lab to industry, lab to clinical, and to industry. So we are ready to support lab to clinical and clinical to industry. That is why I was basically asking Dr. Yao that, okay, how much we are ready, like if today the funds are available, are we ready to start the clinical trial? This is a very big question. So because this is where we are now coming in, and we want, whatever the gaps there are, we have to identify what antigens are available and what are the advancement in antigens are happening. Like there is a very good advancement, and there is a very good discussion we are having on a conjugation, and I always say that, okay, we should focus on conjugate vaccines, because if you see the new vaccine development in Europe and US and Korea, they are working more towards the upper respiratory tract, respiratory infections. Influenza is their major problem. Influenza is not the problem for the third world countries. Third world country, the problem is gastrointestinal tract infections. We have a problem of typhoid, cholera, dysentery. That is our problem. So we have to basically focus, and I always say that we have to focus on that side. When we developed our feasibility, we also know that, okay, human capacity is also not available, like we do not have trained biomanufacturing workforce in Africa. PAVM benchmark, and they said that, okay, they have only 3,000. Am I going out of time? So how much time we have? 5 minutes. Okay. I was thinking that we have basically saving some time so we can spend. Okay. What we did is we want to basically come into all the eco chains. We developed a KINA Foundation, which is a biomanufacturing training center. And today I'm very pleased to say that, okay, we have students of KINA Foundation in this forum. Okay, there are students here which are basically learning. It is a 4-month training course, and after that completing, it's a after graduation you come to do that course, and with the course we are basically helping them, enabling them to get jobs in the biomanufacturing team. Our objective is very clear. We are here and we basically are completing the eco chain at formulation, fill and finish. The first products which are going to come in will be oral cholera vaccine, pneumococcal vaccine, and HPV vaccine. These are basically the initial products which are going to come in, and then the later products will be following. Thank you very much.
Okay, thank you, Farhan. So we'll go over to Atlantic Life Sciences.
My name is Emmanuel Kweku Hayford, and I'm the Quality Assurance and Regulatory Affairs Manager for Atlantic Life Sciences. Well, I also have a video to show, but because of time, I would see how best I can touch on salient areas and then we move on. Because DEK has done a very wonderful presentation with a video, and I thought it would have been in order for me to...
How long is the video?
But we are not here to rub shoulders.
3 minutes. You can show your video.
Then I think I'll show the video so that at least...
Gal doesn't have a video. 3 minutes.
I don't think it's as long as...
We are a little bit ahead of time, so 3 minutes.
So the video can roll now.
Of Ningo Pram Pram in the Greater
I choose
Pram
the video
region,
so that at least you have more pictorial.
Is emerging. This is Atlantic Life Sciences Limited, one of the 2 companies behind Ghana's vaccine manufacturing initiative, where science, innovation, and determination is shaping the future of public health in Ghana. The facility, which sits on a 25-acre parcel of land, has received its first shipment of WHO-certified, already prepared anti-snake serum bulk supply, and production is currently underway in its vaccine production unit, processing the very first batch of trial vaccines for the certification of the Food and Drugs Authority. A serum and a vaccine are both important in healthcare delivery. Whereas vaccines are administered before the onset of a disease, a serum is given as an emergency or immediate treatment of a disease such as a snake bite. In conducting a team of regulatory officials led by the National Vaccine Institute around, we observed that the production of these products is highly controlled and monitored from start to finish.
This is the technology we are using for anti-snake serums. So we are having a technique of lyophilization also in place, and that is very rare. Lyophilizer with us. So you can see the quality of the product also. It should have moisture content less than 3%, and this was our first batch. We are having a moisture content only 1%. So it is well within limit.
Safety and quality assurance, according to the scientists, are paramount. According to them, the rigorous quality assurance protocols in place are to guarantee that every one of the products that comes out of the vaccine facility is safe and effective.
My duty is to make sure what you label as a label claim, the percentage you given as a label claim is actually what is there before it goes to the market.
Operating under the government's One District, One Factory, One D1F program, the $50 million project is hoping to introduce its first range of products onto the Ghanaian market, as well as for the West African region soon.
The trial batches for our national regulator has begun, And once they are complete with all the trial batches, everything will be forwarded onto the FDA for their testing and then approval.
Established this year by a Bill of Parliament, the National Vaccine Institute is mandated to coordinate and supervise research, development and manufacture vaccines in Ghana.
As part of our mandate is also to facilitate the research and development of academia and industry.
As Ghana forges ahead in her quest to be a known vaccine production hub, it comes along with hope, self-reliance and better healthcare for the entire continent.
It feels so good to be a Ghanaian at this time, knowing very well that Ghana has taken up this bold initiative to produce or manufacture her own vaccines for the well-being of her people and also for the rest of the continent. Reporting for GBC News, Gifty Ajay, Pram Pram, Greater Accra region.
So more importantly, I think after the research and development has taken place, there should be that outfit that would make it reach the doorstep of those individuals who have need for all the hard work that is coming from your cortex. And that is why Atlantic Life Sciences would be proudly Ghanaian and would be welcoming you into our fold for all the good works that you do.
We'll talk about that a little bit after, but I'll let Yamachi present. Yao doesn't have a video, but he talks longer than a video, so 5 minutes.
Our video, actually our videos are on YouTube, so if you want to see Yamachi videos, please check out YouTube. We do have them, but I think in the interest of time, Yamachi has already been introduced by Eliza, who is a research scientist at Yamachi. We are slightly different from these two. We are a life science startup, a biotech startup, and so we are almost entirely focused on IP creation. Work at academic institutions like this is the beginning of IP creation, but you typically need an intermediate step, which is a commercial outfit that is focused on research, not yet industrialization, but developing academic ideas to the point where they can be fully realized as products. And so that's the role of biotech, companies like Yamachi. In our case, we are almost entirely focused on oncology. So non-communicable diseases are rapidly rising in our part of the world, and cancer is one of them. And so we think that there's an opportunity to really provide leadership in how cancer is treated and prevented across the African continent. So what we are trying to do at Yamachi is to diversify precision oncology globally by leveraging Africa's genetic diversity. Africa is the most genetically diverse continent, and yet we feature only 3% of global human genomic data. So most genomic data, human genome data available is from people of European ancestry. Almost 80% of it in publicly, or in fact not just publicly, but in generally available databases will be of people of European descent. African people represent only 3% of sequenced genomes. And most of these people would be African Americans who themselves are an admixed population. So given the diversity on the African continent, we believe there's an opportunity to really advance drug discovery in cancer by understanding cancers across Africa. So we're 3 years old. We're headquartered in Abilinkpe. I myself am a research fellow here at WACCBIP and have since sort of begun a migration. So I have 1 foot at WACCBIP and 1 foot, maybe 1.5 feet.
You have 1 toe here.
No, 1 toe. But what we are trying to do is build a viable entity as a commercial business that drives an understanding of cancer, but importantly does so through the creation of IP that can then be advanced through steps all the way towards a new drug. So we are looking really to identify targets, so drug targets that would be helpful in either diagnosing or treating cancer. And the goal is then to license that IP to pharma, who would then take it forward towards clinical trials and ultimately into companies like DEK and Atlantic, who would then manufacture. And so we are part of that ecosystem, and an important one, I think, and one that is not very common in our part of the world, but one that is crucial to advancing research from purely academic enterprise towards translational products that would change people's lives. So we're currently 40 people. Yamachi is now 40 people strong. Almost entire.
I'll mention that we collaborate a lot also on another vaccine preventable cancer, which is HPV. And as Yao said, they are trying to fill the gap in R&D and biotech. And I know that both Atlantic Life Sciences and DEK are more on the manufacturing side. But I think where we can build on our basic science research, which is why we showed the breadth that we did today, and go towards manufacturing, is to do more research and development in collaboration with companies such as yours. For instance, Farhan, you asked questions about, is it ready for the clinic? The National Vaccine Institute is supporting the creation of a sort of a slightly scaled up protein production facility. And I think, so I'd like your thoughts on how your individual companies can basically help to do some of that preclinical scale-up work, maybe in shared studentships. It's a good initiative, the Kina Foundation, but I think what directly impacts, what directions that have you seen, and how do you think you can more directly impact that and push it forward so that we are creating IP between ourselves and ultimately speeding up getting new vaccines to the market, something like what Yao anyway presented earlier on. So I'd like to switch it up a little bit. Okay. I'll let Hayford present, and then Farhan. Yeah.
Who, me?
No, Hayford.
You want me to pick up?
Yes.
Oh, yes, like I earlier indicated, but I did not quite get your question anyway. Because
so
like
we're looking at how can you directly sort of help us facilitate getting, directly or indirectly help us facilitate getting some of our candidates developed, maybe in the scale-up process, in preclinical to clinical.
Well, we have that arrangement, you know, embedded in our business model, right? If you think of scaling up, then of course the point of commercialization also comes in, and that is where you are able to raise the necessary funds, even to support way for you to come to us, you know, sit with us and then discuss how we can be of that help to you. Because you may be coming from an angle where we may not almost immediately see as viable, all right? But then rest assured, we have considered all of these silently and would listen to you. If we are convinced, why not? We can take it up together, and particularly for the fact that we are partnership oriented. When it comes to surviving in this industry, you cannot brush aside partnership. So it will be a welcome idea, and I'll be very quick to tell you that our doors are very much open for some of these discussions. Yeah.
Okay, Farhan.
Thank you very much. Basically, what happened is that Gavi and WHO has already identified the antigens which is necessary for Africa. So working under those antigens, we have seen that, okay, there are so many products which are available, which are in phase 2 and phase 3 and completion of phase 3, which are not localized in Africa. They are basically having, some of them have like Chinese product which have Chinese pharmacopoeial basically specification. So a compatibility study need to be done. So it's not necessary we go back and develop the lab level discussion and we have a lab to lab tech transfer and then toward lab to lab tech transfer, then lab to industry tech transfer. So we have to basically smartly think into, and then what we need to do is the readily available products, we have to localize in Africa before our R&D lab comes in, which is going to be ready in 2026. We can use WACCBIP lab and Nuguchi. where we can do a lab-to-lab tech transfer. Like we are in process of doing that one in one lab. So what happened is that there is a product HPV9 and HPV11. The product is in R&D phase. The problem with the product is that the specification which is used is Chinese specification. So we are bringing it in. We are going to give it to you, and so what you do is you will basically make the product available for us. When it is ready for us, it can be used. So it's a co-development.
Right.
So this is the best approach I think we have understood, and we are doing it in other countries also. Like we have started a R&D lab-to-lab tech transfer in another country. We are also evaluating. We have submitted WACCBIP credentials to 1 or 2 countries. And then once it is finalized, we will be having it. In this way, we can jointly develop the product, and we have like a shared IP, and that shared IP will help Ghana to have its own product in maybe like 2030. Thank you.
Okay, thank you, Farhan. Yeah, anything you want to add to this?
I mean, I think obviously we are slightly different, but I think the Heritage Project is a great example of collaboration between us and WACCBIP. In this case, it was actually a project we initiated and then brought WACCBIP on. It's a project that we got funded by the Gates Foundation. And so in our case, we are an R&D lab. So, the difference between us and an academic institution is probably smaller than the difference between us and a big industrialized commercialization partner. But I think the advantages we give is an ability to be nimble and agile, and to apply a certain amount of focus. Academic labs are involved in training, and so there's a certain pace you have to go when you depend on trainees. Yamachi doesn't depend on trainees, and so we can provide a certain acceleration that is required. In biotech, it's a race. You don't have 5 years to advance the way you would with a PhD project. You need monthly milestones and key performance indicators. So we do basic science, we do basic life science research, similar to what you have in academic labs, but our focus is much more target-oriented, and our timelines are compressed. So it means we don't spread ourselves too thin, we do a few things at a time. But I think the advantage of an academic lab partnering with us is once you get to a point where you think you have something that is potentially commercializable, it's not yet ready to take to a DEK or to an Atlantic. You need to do more work on it and potentially even advance the R&D further. That is where biotech labs come out. We see this in Europe and North America where labs spin out biotech companies. A professor may develop something, get to a certain point, decide, I don't want to stay in the university anymore, I need to spin out a company because that's how I'll advance it. They take the same research, but now they put laser focus on that, hire a few really talented scientists to advance it, and then things move much quicker. So that is the role of a biotech startup. I think Heritage is an interesting project where we are collaborating with WACCBIP, taking advantage of the capacity on both sides. And I think the project will be further along than it would be if either of us worked alone. So I think Yamachi will hopefully not be the only biotech. I mean, there are a few others that are trying to start up in Ghana now. There need to be more. The students in the room should take inspiration from this, and maybe even the faculty, that we need a stable of cutting-edge life science companies. It is not easy, and securing the startup capital is extremely challenging. And some of us are still in that journey. But I think having enough of a base of biotech startups will be crucial to the ecosystem. We cannot just have, the industry partners who are going to mass produce these need an influx of IP that would drive them forward. Currently, they are depending on outsourced IP from China and other places. So ultimately, they advance what we can do in Ghana, but it's still not ours. Their job is not necessarily to develop all the IP. They need us to develop the IP so they have something to manufacture. And so till we can create that bridge, we will always be dependent on IP externally. And I think that is the challenge to those of us who are scientists, is to think beyond the publication, think beyond that Nature paper that you are dreaming about, and identify IP that you can protect, and then develop further.
Okay. So that was related to the question as in, I think we can develop that IP, but I'm wondering what role you've obviously mentioned your role, and Farhan, particularly maybe Atlantic Life Sciences. Do you think you can help in co-developing that IP? Because there's only so far we can take it in basic science right now, in terms of getting to the point where you need more money to scale up. And then I'm going to open it up to the audience for everyone to ask questions. 2 or 3 pharma and biotech reps, as in what do you think they can help to bring your ideas to the market or closer to the market, in terms of not necessarily something commercializable, but that's something that is considered novel IP. So.
Yes, I think one of the things we need to understand from industry is that it is also there to help the academia. As we speak right now, we have a state-of-the-art 120-seater conference room, and it was built with students in mind. And so helping to further develop IPs here and there and all that, there is room for that also. And currently we have brought in experts. And what I mean is that people with that industry exposure when it comes to vaccine manufacturing and SERA in particular. Because at the moment we have one such doctor with us who is there to steer the affairs of, formulations here and there. And the last time I had interactions with him, you could tell that this man is very, very rich. I mean, the knowledge base of this man is very, very rich. And so if you are such a student who is looking forward to understanding anything more apart from what you do, when you come to Atlantic Life Sciences, there are such people who would help or shepherd those thoughts to the right, destination.
Okay, thank you very much. So, any questions, comments from the audience, particularly? Okay, Philip in the back, and then Dr. Charles and Reverend.
Okay, thank you. So my question is, we are trying to do academics. We are doing research in the lab. I wanted to ask what place you have in the 3 different companies for sponsoring students, for instance. I guess I'm coming to direct, and maybe this might not be the right forum to discuss that, but can you sponsor PhD students, co-sponsor PhD students to do research and possibly come up with IPs? I know AstraZeneca, I have friends who their PhD is completely funded by big pharma like AstraZeneca and some other big companies. So do you have a place like that? Can you sponsor 5 WACCBIP PhD students? Hey. 10, 20 if possible. Thank you.
I'll start first because I'm the smallest. So the reason I started Yamachi is exactly that, right? AstraZeneca didn't start off as a giant company, right? AstraZeneca started off small. Roche started off small. The best example is Novo Nordisk. Novo Nordisk is currently one of the most valuable pharma companies in the world. It's about $500 billion. I think it's second only to Eli Lilly. Novo Nordisk alone contributed 30% to Denmark's GDP last year. And it's because of their new drug, Ozempic, which is the metabolism drug that all the celebrities in America are taking to lose weight. But the point I'm trying to make is, this is what Africa needs to do. Your question is sort of the end of the process. So you're asking for companies to sponsor PhDs. In order for companies to sponsor those PhDs, they need money, right? And the way they generate money is by generating products and revenue by commercializing science. So till we commercialize science, we will always be dependent on support from foundations and others that support. WACCBIP was established through World Bank and Wellcome and all those resources. So your question is a good one, but your question is a question actually that should go to, I would argue, should go to leaders of our countries in Africa that why don't we have an AstraZeneca? I just came from ASCO, which is a 40,000 person conference in Chicago for oncology. And I tweeted about, those who follow me on Twitter know I tweeted about it. There was not a single African company there. All the top pharma companies in the world. I mean, Yamachi was there, but I won't count us as, worthy enough to call. Everybody was exhibiting. They had stands bigger than this room. You see Eli Lilly. They were giving out cookies, food. They had fancy coffee. There were trillions of dollars represented in IP in that room. And I couldn't find, at least to my view, a single company. Japan was represented by 6 or 7 companies. Japan is a small country. The continent of Africa was not represented, right? So I hope, right now, Yamachi doesn't have money to sponsor a PhD, right? We do internships for masters, so we started doing something. I hope that in time, yes, I'll be able to sponsor 20 PhD students. Why not, right? I mean, that's why Prof let me do it, right? Prof, and I know he will come to me the moment I have more of these initiatives to provide the resources to support PhDs. So it's a good question, but it's about when do you do that, and why haven't we done it up to this point?
I want to add something here. See, what happened is that this is basically the gap which was there. We cannot wait up to the companies to become so big when we start supporting the students. So that is why we went to GIZ and asked them that, okay, we need the biomanufacturing force now. So GIZ, thank you very much. They have supported us. They have fully funded the project, which is Kina Foundation. Students are sitting down here among you. They are now completing the biomanufacturing training in the 4th month training. They started from March and they are completing in June. They will be now adjusted in the biomanufacturing force of the Africa. Africa needs 9,000 full-time employees. So this is basically you people. One way is directly sponsoring few of the PhDs, and the other way is sponsoring graduates so that they can come and be. So we have already started it. Thank you very much.
Okay, thank you. I believe, Reverend, you had a question? And then Charles.
So my question probably it will be to all of you, but kind of general. Now I'm just wondering, now that you are developing or manufacturing these vaccines, Yamachi, of course, specifically is more focused on cancer. So aside vegetarians, many of us, of course, feed on animals, and I'm just wondering, with the vaccines you are producing, I'm sure they cut across, I just hope, and could be used for veterinary purposes. Because of the quantities you are anticipating to produce, just asking whether they are of use also for vets.
I think I can answer that. Usually there's different companies that will make veterinary products and human products. So because of the different levels in safety and things like that, I mean, they could have done it different lines for those, but I don't think.
It is not possible like to produce in the same even premises the veterinary vaccines and the human vaccines. It is not possible. So you have to build up a separate factory, completely dedicated factory for the vaccines for the human and dedicated factories for the vaccines for the. Even in the humans, you cannot produce BCG, which is a tuberculosis vaccine, in the same factory. You have to. Dedication is important there.
Yeah. So for vets, I think already there's some veterinary vaccine production in Ghana. Is it VSD? So Charles, you had a question.
Yes, I just wanted to, I think before we're talking about building the academic, industry bridge, but in a way too, if you look at a lot of the, clinical trials that are really happening, most of these are taken out of Africa because we don't have the strong regulatory works or probably also, if you think about it, more of the health sector. So we don't have strong system in our hospitals, in our clinics and stuff that can hold some of these clinical trials. It's really, really important. I think probably some of the research institutions, like I probably might commend Noguchi for that, for starting some of these ISO certifications, which is really going on. But how are we bringing our hospitals, our health sectors along in terms of building strong systems to really support this clinical trial? Because that's where the money will come from. If you don't have the strong hospitals with the right system to really hold the clinical trials, we're not going to get them.
Yeah.
Can I take it? Question?
Okay.
See, what happened is, if you talk about the clinical trial and setup of the clinical trial, a clinical trial need a project manager, project management, then overall they need a site manager, site enrollment, clinical researchers. So overall you see that we need basically
That's the last comment.
clinical trial, CROs are important. Here in Ghana, basically we have a setup of clinical trial under the R&D department of Ghana Health Services. We have 4 clinical trial centers. Kintapo is one of the clinical trial centers. So if the clinical trial comes from outside, it can be performed. But initiating a clinical trial in Ghana, we are trying to work out a CRO, so that once we have worked out a CRO, then it will be possible. So right now what happened is that the clinical trial is happening in Ghana for a very long time. Ghana is always participating, but the main researcher or the main lead clinical trial person is outside. So here we have only regional clinical trial coordinator. So that is what we are. Here is a lady, I think, sitting down here whose name is Clara, and she is our basically clinical trial coordinator. We have already appointed and we are working with it. Thank you.
So, well, Farhana said it, but it's not correct that we have a weak clinical trial. We have a very strong clinical trial infrastructure. We have a strong regulator. The FDA has been regulating clinical trials in Ghana. And as he said, the Ghana Health Service actually has, various clinical trials in different health research centers in Ghana. We just did a COVID-19 effectiveness study with Kintampo. So, we don't hear much about the clinical trials, and so that's something that the Vaccine Institute is taking as a key activity, working together with the Ghana Health Service and the FDA to strengthen the clinical trials infrastructure in Ghana, not just for vaccines, but of course the FDA looks at drugs and new investigational products. There's actually even a traditional medicines directorate, that looks at herbal products, etc. So sometimes we, the academics, we think that because we are not involved in certain activities, it means that it's not that strong. But we probably, that's another aspect that we need to do. We need to make the information available so that people know the strength of our local system and our ability to move things forward. In fact, one thing that we're going to be doing together with WHO is to come out with what we call preferred product profiles for vaccines that we will be developing in Ghana based on the international WHO, recommendations for preferred product profiles for vaccines. So these are things that are ongoing. So your question is good in the way that, yes, there is some work ongoing, and we need to highlight, that, and also make those sources of information available so that people know what is going on. It's not every clinic and hospital that can do a clinical trial, by the way. So, clinical trials are specialized things. And if you go to KNUST, they have a certain center that are set up and have been doing some clinical trials, even in the Asante Akim area and Agogo Hospital and other centers for some time. So just to show that there is a strong clinical trial infrastructure in Ghana. Peter, can we wrap up? It's 1 o'clock.
Yes. So we are exactly at 1 o'clock. I'd like to also recognize the presence of the country technical lead for the RISA Fund, Gameli, who's sitting in the middle there, and also Dr. Amu from CSIR, and a representative of the Armed Forces Health Directorate, sitting just in front of Dr. Amu. And if I didn't call your name, all the deans, directors, you are recognized. And I'll call on the chair for his closing remarks. Thank you very much to our panelists.
So it's been a very wonderful morning. I think we've talked a lot about research. I have my colleagues from the Vaccine Institute at the back there somewhere. Where are you? Vaccine Institute. Are you the only one? In front, too. Yes. I asked them to come because when I talk about R&D, sometimes the auditor will ask me, Prof, are you sure that you are using the money well? And then when I asked the chief accountant to sign a check, she wants to know exactly where the money is going. So I asked them to come to, please sit down. I asked them to come to this so they understand, what the end product is going to be. When I say I'm supporting WACCBIP, if I want to give some PhD or some master's students some allowance, they'll understand where we are trying to go. And then as for DEK and Atlantic Sciences, you've seen the videos you heard, so you know exactly what they're about. So we had a wonderful time. I'd like to thank GIZ for supporting us, and Professor Wandari and WACCBIP, for allowing us to work together with you so that we can actually make vaccines in Ghana. So from the MVI, thank you so much.
All right. So I think it's been a fantastic day. Wouldn't you agree? I think next time we'll make it a full day. Yeah? But once again, I want to thank all of you for taking the time to join us. I want to specifically thank our friends from GIZ and the National Vaccine Institute for leading this initiative to have this program. We hope that this is the first in a series. We should be doing it every other month or something like that, and keeping abreast with what each of us is doing, because we need to work together to move the country forward in terms of our scientific independence. Now we have industry, as we've seen, from biotech to manufacturing. Like Yao mentioned, we have that deficit, that gap in the biotech capacity, which is needed to take the knowledge that we generate here in the academic institutions and translate that to a product that can be manufactured. So working with the Vaccine Institute and our partners, we hope that we'll be able to close that gap and allow the full potential of vaccine development in Ghana to be unleashed. There are so many excellent ideas that have been generated in the academic institutions. And if we can have that capacity to move those forward, it will be very exciting. We all know that it's not every vaccine candidate that would be successful. Indeed, less than 5% of them would be successful. But if you don't try, you wouldn't know, right? You have to try and see which ones would work. And we need to have the system for doing that. And once we hit, it'll pay for itself, right? If you get one vaccine, it'll pay for all the PhDs that we've been paying for, yeah? So let's all recommit ourselves and continue to carry forward with the passion that we've all shown. And I'm very confident that vaccines will be coming from Ghana very soon. So I thank you all for your time. We are happy that you visited us. Please, if you've never been to WACCBIP, you can walk around and look at the labs and, meet our students and faculty and staff who are in the building. And welcome back anytime. Thank you very much.
It's lunchtime.
So we are done. If the lunch for as before, for the invited guests, will be on the first floor, and for everyone else on the second floor. Yes.